Okay, Assalamualaikum. Good evening, selamat petang. This is lecture number 7 for SII1002 of surface processes. In the previous lectures, we've looked at sediments, sedimentary rocks, the process of transport, weathering, deposition, and also the process of lithification, where you change sediments into sedimentary rock, okay, which is covered under diagenesis. We've also looked at classification of sediments and sedimentary rock. So you have chemical sediments and sedimentary rock, and also classic sediments and sedimentary rock. We've looked at the textural properties of classic sediments and sedimentary rocks also. Okay, so today our topic is fossils. So let's have a look at what fossils are. Okay, so what are fossils? Right? Fossils are the remains of or traces of ancient organisms that lived in the remote past. Okay, so maybe you have an animal, let's say, and this animal has a skeleton, an exoskeleton or an endoskeleton within, right? Or a shell, and the animal dies, and you're just left with the remains, right? It's not. For example, here I have the skeleton of a dinosaur, so that's a fossil which has been preserved. Huh? Also things like this is the exoskeleton of a trilobite, a once living organism. So that is also a fossil. Okay. Now the study of fossils is called paleontology, right? And yeah, someone who studies fossils is called a paleontologist. Now, fossils. Where can we find fossils? What types of rocks do you actually can you actually collect fossils from? Well, fossils are preserved in sedimentary rocks. So remember this. So you can find them in rocks such as mudstones, shales, siltstones, sandstones, or limestones. You don't find fossils in igneous rock because igneous rock originally was molten at very high temperatures and pressures, and then was cooled and crystallized, right? So you think about living organisms, we're not going to live in environments which are thousands of degrees Celsius or several hundreds of degrees Celsius. So most organisms are living on the surface of the earth, so at very fairly low temperatures, and yeah, most of them are living on a sedimentary substrate. Okay, so for example here we have some fossils which have been collected from an outcrop in Perlis in northwest Peninsula Malaysia. So that's the outcrop. You can actually see some paleontologists working on the fossil site there and collecting these specimens here, right? So in this case, the host rock, the rock which is hosting the fossils, is a mudstone, right? So here we have fossils like this trilobite here, a fossil ammonoid, which is a relative to octopuses, but you have it has a hard shell. In this case, it is a coiled shell. You also have fossil shelly organisms like bivalves, which are still around today, right? Things like cockles and oysters and clams, and brachiopods. So quite a variety of fossils are found in this fossil site here, and the host is a mudstone. The original environment was actually a shallow sea, and the seafloor was very muddy, right? And then all these fossils were buried, all these organisms were buried in the very soft, muddy substrate, and then the mud is compacted, is lithified to form mudstone, and after several millions of years, exactly 340 million years, the rocks are exposed as mudstone. Okay. Just to show you one more photo here, these are also fossils. These are fossil unicellular organisms. These are foraminifera. They have a skeleton made up of calcium carbonate, so they are easily preserved. In this case, they are preserved in a sandstone. So this is the sandstone bed. We are actually looking at the bedding plane of the sandstone. You can actually even see some ripples, symmetrical ripples on top of it. So you had waves. So we know that it was a shallow sea, something like 270 million years ago. This was during the Permian, and you had microorganisms living there with a calcareous skeleton, and then they were buried, they were lithified, and now preserved as fossils. Okay, so these are fossils preserved in sandstone. You can find a lot of fossils in limestone because a lot of limestones throughout the whole world, including in Malaysia, they have a mainly biogenic origin. Yeah, just made up of lots and lots of skeletons of dead animals, right? For example, you have coral reefs, you have shelly bivalves, brachiopods with teeth of fish, starfish, and so on, right? So all these have hard parts, and they accumulate to form the rock limestone, right? For example, these are limestones from northwest Peninsula Malaysia. This is from Perlis. This is from Langkawi, and you notice these spots, light-colored spots here. These are actually fossils of echinoderms, relatives to modern-day sea lilies, and also starfish, right? And these are fossil shells of, again, the relatives to squids. Right? So they were ancient mollusks. So we call them nautiloids in this case, right? So all these specimens are actually from Malaysia. So fossils are not really that rare. If you know how to find them, you know which type of rock you need to search for, search in, right? So try to find sedimentary rocks. Most likely you'll get fossils out of them. Now, but it's not that all sedimentary rocks will have fossils. You need to have the right conditions for the process of fossil making, or process of fossilization. There are two main factors affecting fossilization. One, it is best to have, for the animal is dead, but yeah, for fossil preservation, it is best to have hard parts. The organism needs to have hard parts. Either it's a shell, an outer shell, an exoskeleton, or bones, which is an internal skeleton, right, an endoskeleton, or teeth. Teeth are very easy to preserve. Why hard parts? Well, they're hard, they're very resistant to erosion, right, and hard to destroy. So you tend to get a lot of hard parts being preserved in the fossil record, especially teeth. For example, the mammal fossil record is, you have a lot of the record is mainly made up of lots and lots of teeth being preserved in the rock record. Okay. Now, another factor that can affect fossilization is the rate of burial. Right. You need to bury your carcass, the living or the remains of your once living organism, very fast. Okay, in order to stop decay, right? You don't want to have bacteria attacking it, right? To hasten the rate of decay, right? Increase rate of decay, right? So you need to have rapid burial. For example, maybe you have a dinosaur, with soft tissue, and then it's been swept by a flood directly into a river, right? Into a river channel. Then it's buried by thick sand and mud. So those are very good conditions for fossil preservation, right? It stops fast decay of the soft tissue. You can even preserve soft tissue in that case, right? So rapid burial is a good is a factor that can help in the preservation of your fossils. Okay, so a combination of rapid burial and the possession of hard parts is ideal. So for example, a clam which is buried in thick deltaic sediments will have a higher chance of being preserved compared to the remains of a jellyfish with no hard parts, right? But sometimes you can get the preservation of organisms with no hard parts in the rock record. For example, there are jellyfish fossils preserved in shales, and these have been reported, right? How does that happen? Well, even though it doesn't have a hard shell, it is buried very rapidly. So you stop the decay, right? Okay, so it's good to have hard parts and then rapid burial to increase the chances of your remains to be preserved in the rock record, to become fossilized. Now, let's have a look at the ways that fossils can form. What are the modes of preservation of body fossils, right? So here is a list of the common modes of preservation of body fossils. You can have unaltered remains, you can have replaced remains, or permineralized remains. You can have also recrystallized remains. You can have fossils preserved as molds and casts. These are acuan in Malay, right? Or just preserved as carbon films, or this is carbonization. Okay, so let's have a look at these one by one. Apologies, I need some water so my mouth is dry. Okay, so let's have a look at the different modes of preservation, of fossil preservation, one by one. Okay, so the first type, the first mode, these are unaltered remains. So what is meant by that? You preserve the actual biological remains. Right? For example, here we have a baby mammoth which has been fossilized, right? And the baby mammoth here is 40,000 years old, buried in frozen earth, permafrost, okay, in cold climates, right? So it still preserves soft tissue. You can still see skin, hair, and fur, right? You can still see the earlobes and the trunk, and also the lips and the eyelids, right? So these are soft tissues which rarely preserve and usually tend to decompose very rapidly, right? So in some cases, even the stomach content was preserved, so we know what they ate before they died, right? So how does this happen? In this case, the animal was frozen, right, in a cold climate, right? So this resulted in a slower rate of decay, right? So like you put some some meat into the freezer, right, in your fridge, right? But this thing has been in the freezer for 40,000 years, right? So it doesn't have to be just simple freezing. It could be the drying up of your carcass, that is mummification, right? You can also encase animals in amber, which is this is fossilized resin from a tree, right? So the animal got stuck in the resin, right? And in this case, you have a fossil spider being preserved in fine detail. You can even see the hairs on its legs here, right? And also a fossil insect here. And this is 100 million years old, right? During the time of the dinosaurs, right? Or in other cases, you can even have animals which fell into tar pits, right? Petroleum-containing sediments, and they are encased in that. So again, that disrupted the decay of the soft tissue, right? So that's the first type, unaltered remains. The actual tissue of the once living organism was there. But in other cases, you have what is called replacement. This is where the original mineral of the skeleton has been dissolved and then replaced by a different mineral. Okay, so this is diagenesis, right? So it's changing the mineralogy. Maybe there was water, groundwater percolating through the rock, bringing it with ions. You get chemical reactions. So you change the chemical composition of your fossils, right? For example, here I have bivalve fossils here, and we still have bivalves around today, right? Like clams and oysters. And we know that modern day clams and oysters have a shell made up of calcium carbonate, SII1002, in the mineral aragonite, right? But here, this fossil here, which is 340 million years old, has this yellowish color, metallic luster to it, right? So it's made up of the mineral pyrite, which is iron sulfide, right? So in this case, the original carbonate shell has been replaced by the mineral pyrite because of chemical reactions between the groundwater with the sediment, and also because of bacterial activity in the ground, in the sediment. Okay. This is another example of pyrite replacement. Originally it was calcium carbonate, now changed to iron sulfide. This one is very nice one, very pretty one, right? Of an ammonoid shell, so it's been replaced by pyrite. Okay, but it doesn't have to be pyrite. Any other kind of mineral. For example, here another ammonoid, which is a mollusk with a shell, a coiled shell in this case, but the shell is no longer calcium carbonate. It has been replaced by silica, SiO2, the mineral quartz in this case. Okay, so that's replacement of the remains. So another mode of preservation another mode of fossilization is permineralization. Okay, So you have permineralization. Yeah, you get permineralized remains. What is permineralization? This is where you have in some remains of organisms, you can have a lot of pores, right? A lot of empty space inside it, inside the skeleton, inside the bone, maybe. Or for example, if you have a tree, it's made up of wood, right? And we've learned in secondary school biology, if you put the wood under a microscope, this is what you will see. You see cells with walls, right, around them, right? The resistant walls, right? So this is what you see. So when the tree dies, all this organic material inside the pores here, they're long gone, have decayed or moved somewhere else. You are left with lots and lots of empty spaces, your pores. You have significant porosity in this case. And then water, groundwater, can flow through these pores, bring with it ions, and sometimes the ions can be precipitated given the right conditions, right? And in this case, silica is crystallized inside the pores here. Silica entering the cell voids in the fossil wood. So you get petrified wood. Okay, so this kind of process, permineralization, because it's impregnating into the smaller scale holes and spaces in your skeleton here, in this case of wood, right? So you can actually preserve really fine details, right? This is a cross section of fossilized wood, right? A petrified wood. You can see the tree rings being preserved. So these are annual rings, right? It doesn't have to be wood. It can also be things like bones of animals, like bones like dinosaurs or fish or mammals, right? So they can also be permineralized as water flows through the water flows through the spaces for nerves and blood vessels, right? And minerals can grow inside these pores, right? So yeah, it's a process of fossilization that occurs when an organism is buried. The empty spaces within an organism becomes filled with mineral-rich groundwater. So the minerals precipitate from the groundwater, so they occupy the empty spaces. This process can occur in very small spaces, such as within the cell wall of a plant cell. Small-scale permineralization can also produce very detailed fossils. Okay, but it's not. There are also modes of preservation, modes of fossilization, which do not preserve the small-scale features. For example, recrystallization. So what happens during recrystallization? You still get groundwater flowing through the pores, right? And you have chemical reactions. So what happens is that the shell material reorganizes. For example, you might have started with a shelled animal like this, right? So this is an ostracod. It's a small crustacean with two shells, two hard parts, right? So these are the two shells. Now, the original material has been changed in this case. First, originally this was the mineral aragonite, which is calcium carbonate, and it was fine grained, right? It had very small crystals forming the shell, right? But then you have recrystallization. What happened is that the mineral recrystallizes, rearranges itself to change into another form of calcium carbonate. It changes into the mineral calcite, which has a different crystal structure. And in this case, the crystals growing are much larger compared to the original material. So this is recrystallization. Now the thing about recrystallization is that it doesn't preserve the finer features because all that has been rearranged, right? So you don't really see the finer anatomy to it. Right? Maybe you have laminae here, the growth lines, they are not preserved. Okay, so it's very crude form of fossilization. Okay, so the original fine structure of this fossil is destroyed in this case. Then you can have this mode of fossilization that we call impression. So you are just left with an imprint of the once living organism, right? Sometimes an organism will leave an imprint in sediment. Like for example, you take your hand and you just stick it into soft mud or maybe clay or Play-Doh, right? So you get an impression, right? So that is an impression, right? And if that is preserved for millions of years, that is a fossil impression of you, basically, right? So if that imprint is either rapidly buried or left undisturbed during slow burial, it can be lithified and become a fossil. So how does it happen? So let's say you have this shelly animal, in this case a trilobite. It falls down onto soft substrate, into mud. It's buried by more mud, right? And you get that surface imprint of that organism. And then the original skeleton is dissolved, yeah, it's destroyed. You're just left with an empty space, right? Right? But then sediment can infill the empty space, and this is what you get. You get a cast of the impression, right? So this negative relief here, the impression of the outer surface of the animal here, that's the mold, and the infilling of that mold is the cast, right? It's like kuih bahulu lah, if you do it right, make that impression, right? Okay, so yeah, deeper imprints are called molds. So these are molds. If a mold later fills with sediment or minerals, it will form a copy of the original fossil, and we call it a cast. Okay. So in this case, another example, fossil ammonoid, this is the mold, and this is the cast, which has been formed as minerals infilled the mold. Okay. Alright. Then, another mode of preservation is what we call carbonization. I think this is the last one. Yeah, this is the last one. This is when the soft parts of an organism have been compressed, they are heated. This can happen when you bury it very deep, right? So the deeper you go into the earth, the hotter it becomes, right? Okay, so it becomes compressed, it becomes heated. This heating process, basically you are cooking the organism, right? The remains of the organism. So it drives off all the volatiles, things like hydrogen, nitrogen, oxygen, and what is left is just a thin film of carbon on the bedding plane of your rock. Okay, so a carbon film is left behind. This usually happens with fossil plants, right? For example, here these are all fossil plants. So all the volatiles have been driven off to somewhere else. Because of the high temperatures, you are just left with a thin carbon film. But even though it's just a carbon film, lots of details can still be preserved. For example, you can still see the fine venation of the leaf here, right? And these are fossil ferns, right? This is from Pahang, in near to, if I remember, this is near to Jerantut. You can still see the stem, right? And also the small leaves here, and sometimes even the spores, right? So this is fossil carbonization. It's common in plant material, but you can also preserve fossils through this process. For example, organisms with phosphate skeletons, organisms with chitin skeletons, for example, insects can be preserved through carbonization, and fish can also be preserved as carbon films along bedding plates. Okay. All right. So that's the first part of the lecture. We've looked at what fossils are, where can you find fossils, and the modes of preservation of fossils. Okay. So for the second part of the lecture, we're going to have a look at the applications of fossils, because in geology, fossils are very useful, especially if you are doing things like geological mapping, trying to find resources, right? So a lot of geologists actually use paleontology to find earth resources like hydrocarbons and so on, right? So this is a list of the main uses of fossils. One, we use them for the relative dating of rocks in geological events. We use fossils for the correlation between separated rock units. This is very useful in search for economic minerals. We use fossils to interpret the ancient environment, ancient paleo environments, or to interpret paleoclimate. Fossils were the main evidence for plate tectonics during the early days. And of course, very academically, intellectually interesting if you are curious and want to know more about the history of life, how it changes through time. Okay. So let's have a look at how fossils are used to know the age of rocks, right? How do we know the age of rocks? For example, this is a geological map of Langkawi. Right? So based on studies on fossils and rocks, we know a few things about the history of Langkawi. For example, the Machinchang Formation along the northwestern coast of Langkawi is made of sandstones, and they are actually the sandstones are quite old. They are the oldest rocks in Malaysia. They're something like 500 million years old, right? And you also have trilobite fossils inside these sandstones here, right? And we know that the limestones at the Kilim area along the eastern coast of Malaysia, these are a little bit younger, something like 420 million years old, right? And then you have really young rocks in the middle, the area of Gunung Raya here, right? It's made up of granite, and actually know that it is something like 225 million years old. But how do we know the age of rocks? So that's the next part of the lecture. How do we determine the age of rocks? The first method is called relative dating. This is determining the age of rocks based on fossils, right? But it is a relative kind of method, right? We know that rock can be older or younger to our reference. That's the best that we can do. Right? But you also have absolute dating, where now you can put a number to it, right? 500 million years, and so on, right? So this is called absolute dating, and it's based on the principle of radioactive decay of elements, right? For example, uranium, potassium, rubidium inside the rocks. Let's have a look at relative dating first, right? How did the geologists determine the age of rocks before the advent of the atom bomb, basically, right? Before nuclear physics, right? So in the old days, we have lots and lots of geologists. We're doing mapping, we're describing the rocks, describing the fossils that are found in the rocks. They found a few things. They observed a few things. First, the sedimentary rock layers are organized into vertical successions, right? It's not random. And each layer contains its own distinct fossil assemblage. And there was a sequence of organisms in the fossil record. This is called the principle of faunal succession. For example, if you find fossil trilobites, they are always in the oldest beds, near to the bottom of the succession, wherever you find them throughout the whole world. If you find dinosaur bones, they're usually in the middle of the rock succession. And mammals become more common in the topmost beds. Okay? And this is based on observations by hundreds or maybe even thousands of geologists working throughout the whole world and publishing work, presenting their finds in conferences, right? And we come up with this understanding here, right? Okay, so that's the principle of faunal succession. Biota have followed one another in an orderly succession through geologic time. Okay, so all these paleontologists looking at fossils, looking at the rock record, they've looked at all these fossil assemblages. So they've arranged all these fossil assemblages to form a history of the Earth. This table is called the geologic time scale. Okay. So this shows the history of the Earth, and the data is mainly based on fossils, right? So the history of the Earth the time scale can be divided to several units that we call eons. There's Hadean, there's Hadean, Archean, and also Proterozoic eons. And also you have the Phanerozoic eon. Okay? And so going from older at the bottom to younger at the top, okay? Usually what we do is that we lump together Proterozoic, Archean, and Hadean into this group that we call the Precambrian, so before the Cambrian, okay? But yeah, then you have, but there's more focus on the last eon, the youngest eon. This is the Phanerozoic eon. Why do we focus more on this? Because most of the fossils preserved are in the Phanerozoic eon. Fossils are relatively rare in the Precambrian. Okay, and then you can subdivide the Phanerozoic eon into several eras. You have the Paleozoic, which is the oldest era, followed by the Mesozoic, and then the Cainozoic. All right, and then you can subdivide these eras into smaller periods. Right? So for the Paleozoic, the oldest one, is divided into Cambrian, Ordovician, Silurian, Devonian, Carboniferous, and Permian. The Mesozoic is subdivided into three periods. You have the Triassic, followed by Jurassic, and then the Cretaceous. Right. So Mesozoic is the time of the reptiles. You have the dinosaurs dominating the Earth during this time, right? So that's why you have Jurassic Park, right? And then the age of mammals is called Mesozoic. It's divided to several periods also. You have the Paleogene, Neogene, and also the Quaternary. Okay. There's also a smaller scale subdivision. We'll just focus on the Cainozoic here. Into Paleocene, Eocene, Oligocene for the Paleogene, Miocene and Pliocene for the Neogene, and also Pleistocene and Holocene for the Quaternary. Right. So we are in the here. Right. Okay. So this is one advice. You need to memorize this. Okay, the geologic time scale, because you are going to use it until final year and also in your career as a geologist later on, right? So you should know the Precambrian, followed by Phanerozoic, which is subdivided into Paleo, Meso, and Cainozoic. You should know the periods: Cambrian, Ordovician, Silurian, Devonian, Carboniferous, Permian, Triassic, Jurassic, Cretaceous for the Mesozoic, Paleogene, Neogene, Quaternary for the Cainozoic, and also the subdivisions into epochs for the Cainozoic. Okay, so a little bit of rote learning, but this you really need to know. Okay, so yeah, there is a distinct fossil assemblages for different parts of the geologic time scale. For example, like I said, in the Precambrian, Proterozoic, Archaean, you don't have multicellular fossils. You only have things like unicellular bacteria fossils. You have fossils of the oldest fungi, right? And during that time, even the oldest rocks, the Hadean, there are no fossils preserved. Right? But then in Paleozoic, you start to get invertebrate fossils. Invertebrate fossils are very common, especially in marine sediments. You get lots of marine arthropods, for example. You get evidence of the oldest forests occurring in the Devonian, right? And land vertebrates, amphibians, and reptiles started to diversify during the Carboniferous and Permian. And the Mesozoic is the age of reptiles. You have dinosaurs, pterosaurs, and the first birds rising during that time. Okay, and then you have the Cainozoic. This is the time of the mammals. Lots of mammal fossils during this time. So they reign supreme on land during that time. Okay, so different ages have different fossil types, fossil assemblages. Yeah. So this is so going back to that principle, there are different rock layers. We have different fossil assemblages. This is a very useful thing to know, right? Say for example, you have 3 exposures of rock. They are separated by large distances. In this case, these 3 units here are separated by several kilometers, right? Right. They were originally continuous, but because of erosion, they are now separated into 3 hills. Right. So let's say in this unit, I'll take the topmost part. Okay, unit E here, right? This unit E here, on this side here, you have this kind of brachiopod. Okay, so it's common in these rocks here, right? And there is a certain resource found inside these rocks, and let's say a certain mineral, right? And you are interested in trying to exploit that mineral. Okay, so if you go to another location, you should find the minerals also in these layers because it also contains the same fossils, and so on, the same fossils, right? Okay, so we make the interpretation that these are equivalent in age. They were deposited during the same age. Okay, and so this exercise that we're doing here, like we're trying to see how far it goes here, try to connect these different layers here, even though they have been separated. It's called correlation. Okay, we establish a time equivalency between the layers, between the strata, right? So in this case, you have more fossils, so you get more accurate correlation, right? So here, trilobites bottom followed by this type. It's got the same kind of pattern, so you know where to connect your lines. Okay. So fossils have proved to be the only widely applicable tool of time correlation in sedimentary rocks. Fossils are, well, it's cheap to do. You don't really need to have any advanced equipment, right? Just need to find the fossils. That takes some time and effort to find those, and you can now determine time equivalency between layers, right? And in some cases, fossils can be fascist independent. They do not follow the type of the rock. For example, this fossil here, in this case, is found in a sandstone. But if you trace it to another area, is found in the mudstone, but we know that it is the same age because this fossil is only found during a certain time period. Okay. Now, especially useful for correlation is what we call index fossils. These are specific kinds of fossil species or genera which are used to define and identify geologic periods or faunal stages. Right? They work on the premise that although different sediments may look different depending on the conditions under which they were laid down, they may include the remains of the same species of fossils. For example, if I find the trilobite species Paradoxides pinus anywhere throughout the old world, I know it is in the oldest layers in the Cambrian period. Okay, so because it is an index fossil, we know that it is only found there. Okay. Okay, so yeah, so the different fossils are confined to rocks at different ages, and then we construct a relative geologic time scale for the history of the Earth using the fossil assemblages, right? Yeah, just a nicer picture with lots of photos here. I forget where I got it. Apologies. Okay, so very useful for relative dating of rocks and geological events. Useful for correlating between separated rock units also, right? Yeah, and you can determine the age of rocks relatively. You just know whether it's older or younger. But what about if you want numbers? So here comes along absolute dating. So we need to do absolute dating, which is based on radioactive decay of elements. I think everybody here has learned in secondary school regarding radioactive decay. I'm not a specialist, I'm not a physicist. Just provide a simple explanation here, right? So let's say you have elements in a rock. A lot of time it is an igneous rock. So yeah, we know that elements may exist in different isotopes. Okay, so each isotope of element, we refer to them as nuclides. So different nuclides, they have different numbers of neutrons in the nucleus. Okay, so some nuclides are unstable. They tend to change into a daughter nuclide, another nuclide, right? Through radioactive decay, right? So the collection of these atoms here of a radioactive nuclide, it will decay into the daughter nuclide, and the rate is exponential. Okay, and that rate is called the half-life. Okay, so after one half-life has elapsed, one half of the atoms of the nuclide will have decayed into the daughter nuclide. So for example here, you started with an element A, isotope A here, right? And so that's the amount, the total of isotope A in this case. But isotope A tends to decay into isotope B. Sorry, yeah, I just need to remember in this case. Where's B? B here. Okay, so that's this dashed line here. So after a certain amount of time, one half-life, let's say, right? Let's say, no, that doesn't matter right now. So let's say 500 million years. That's one half-life. So the amount of the material A has now reduced to half of the original amount, and because of half-life. Okay, and half of it has changed into the daughter nuclide here, which is material number B. Okay, so another 500 million years, another half-life. Now it's reduced to a quarter, and so on and so on. So you get an exponential rate of change from the parent nuclide to the daughter nuclide, okay, through radioactive decay. That rate is the half-life. So we can use this because it has an exponential rate, right? Every half-life and so on, right? There's a rate to it. We can use it like a clock, right now. Okay, so we people are using it for radiometric dating. This concept of radioactive decay, right? Radiometric dating, often called radioactive dating, is a technique used to date materials such as rocks, usually based on a comparison between the observed abundance of a naturally occurring radioisotope and its decay products. So for example, if you start off with 100, let's say use the term units here, uranium-235, it will halve into 50 units of uranium-235. and the other half is going to has changed into lead-207 after its half-life of 0.7 billion years. Okay, so these are some of the isotopes that are used for radiometric dating, including uranium lead decay is used for dating, potassium argon, rubidium strontium. Okay, notice the half-lives here: 4.5 billion years, 1.3 billion years, 47 billion years. So they are suitable to date rocks which are very old, right? But usually in the news, you go to news, people are always talking about carbon dating, right? Carbon 14, for example, in this case, right? But in geology, we don't use carbon 14 to date rocks. Why? Notice the half-life here. It is only something like 6,000 years. It's too short to date rocks which are millions of years old, hundreds of millions of years, or even billions of years old. Okay. So yeah, where do you find these radioactive elements? Here, they're usually stored in minerals and other materials inside the rock. Minerals like zircon, uraninite, biotite, muscovite, and usually, yeah, and these are usually associated with igneous rocks, right, from volcanic rocks, for example. So now we have a method to actually determine the age of the rocks through numbers, right? Absolute dating. Okay. So let's say, for example, we have a sedimentary rock succession like this. So these are the oldest rocks, and these are the youngest at the top, right? And then we have fossils inside them. So we can determine relative dating. These are older rocks here with this fossil. These are the younger rocks here with these fossils here. But in this case, we are fortunate that we have two layers of volcanic ash which have been deposited after a volcanic eruption. And we do radiometric dating there, we get ages. So in this case, we know this volcanic ash is 151 million years. This ash here is 140 million years old. Okay, so now we have a more intellectually satisfying story to tell, right? So we can say, in the beginning, we had a mudstone at the bottom being deposited, and fossil A proliferated during this time. But then there was a volcanic ash, and fossil A became extinct, followed by more deposition, and this time we had a new fossil forming, right, after 151 million years ago. But this also disappeared at 140 million years ago, followed by another volcanic eruption, and more sedimentation at the top. Okay. So yeah, so we have a combination of relative dating and also absolute dating to come up with a better understanding of the history of the Earth. Okay. So this is the final product. That's the geologic timescale. So you had your eons, your eras, your periods, and your epochs here, right, based on the fossil composition. But now we can actually put numbers to the ages here, to these periods and eons and eras and so on. So the Cambrian started 540 million years ago. So the first multicellular life forms originated somewhere around here, 541 million years ago. Right. The extinction of the dinosaurs happened 66 million years ago, at the end of the Mesozoic, end of the Cretaceous. Right. The first forests with plants and trees developed in the Devonian, and we know the ages now. This didn't put here. You can learn more details later in other courses, paleontology, for example. Okay. So we can determine the age of the rocks using a combination of absolute dating and relative dating. Okay. You can do correlation. You can also interpret the paleo environment. For example, if you find fossil fish have gills, they live underwater. So we can interpret that those rocks, for example, this shale here was deposited in a lake, and so on, right? For example, we have dinosaurs, and these had nostrils and lungs like us, and legs, so they were terrestrial animals. So the environment was continental, right? If you find corals, the environment was marine, and so on, right? Yeah, just to show you some photos. This is actually from Kedah, in Binjol in Kedah. You can get these Triassic ammonoids preserved in shale, right? So Binjol is now continental, it's on land, right? There's a road there and just an outcropping of rock. But in the Triassic, something like 230 million years ago, Binjol was underwater, right? because there was a shallow sea there during that time. Okay. Another thing that fossils can tell you is paleoclimate. How was the climate during that time? Because we know that some organisms prefer to live in certain habitats, in certain climates, right? So the paleoclimate of a given region during a certain time can be determined based on the types of fossils and their morphologies. For example, modern day tropics in Southeast Asia, if you go to a riverbank, a muddy riverbank, you tend to get this tree here, the Naipa palm, pokok nipah, right? Naipa fruticans. And it has these fronds here, and it bears fruits with seeds and so on, right? Okay. So it lives in the tropics today. It just lives in the tropics. It doesn't live anywhere else. But then you go to the UK, you find in old sedimentary rocks, which are 56 to 34 million years old, you get the remains of the Naipa palm, right? In this case, this is the seed of a Naipa, seed and fruit of a Naipa palm here, right? But it is 56 million years old, right? So we can make the interpretation that 56 to 34 million years ago, in the UK, in Britain, it was warm tropical climates. Okay, so that's how we make those kinds of interpretations. So I mentioned earlier that fossils were one of the earliest supporting evidence for the theory of plate tectonics, right? We've learned that continents are drifting, drifted also during the past, right? And they were colliding with each other, separating from each other, and sliding each other. So the Earth is dynamic, right? It's not static. The continents can change position, change latitude and longitude. That's why Britain was in the tropics millions of years ago, right? Because of plate tectonics. Okay. So you can use this to you can use fossils to reconstruct the paleogeography of these tectonic plates millions of years ago. For example, in many places throughout the whole world, you get this long-shaped leaf plant that we call Glossopteris. Okay, it is a gymnosperm, right? Found in all the southern continents. It's found in South America, Africa, Antarctica, Australia, India, and Madagascar. Today, all these are separated by large oceans, right? They are far away from each other. But we make the inference that about 299 million years ago, all these continents were near to each other because they shared the same flora, they have the same plants there, right? Fossils of the gymnosperm Glossopteris found in all the southern continents provide strong evidence that the continents were once amalgamated to a supercontinent, large supercontinent, and during the Permian. Okay, it's called Gondwana. Okay, so it's not just fossil plants. Other land organisms like mammal-like reptiles have been found crossing continents, right? Found in South America, Africa, and other kinds of mammal-like reptiles, also not just cyanoids. And this provides evidence that they were actually once attached to each other millions of years ago. Okay, so I'll stop there for this lecture, right? So we looked at fossils. What are fossils? How they are preserved, where are they preserved, and what are they useful for, right? Okay, so I'll stop there. Thank you, and have a good evening.